왜 내 학교에서 왔어?
Good afternoon and nice to meet you everyone. My name is Juwon, PhD student from Daegu University in South Korea. First of all, thank you for the opportunity to give a talk today. For today's talk, I'm going to present my master's thesis. The title is Genetic Diversity and Structure of Invasive Deuteria using serial sequencing. Here's the outline. My talk has 4 parts. I start with backgrounds of the study, then move to materials and methods. After that, I present the result and finally a discussion. Let's start with study species. Deuteria, the scientific name is Myocastor foipus. The species belongs to the order Rolitia and the family Echinidae and is native to South America. It's the semi-aquatic rodent that breeds twice or 3 times a year. The species reproduces quite a lot like any other rodent species. On average, the species pairs 4 pups at a time, but in some cases, up to 13 pups in a single reproduction. Deuteria is listed by the IES2026 as one of the 100 worst invasive alien species. In Korea, since its initial introduction in 1985 for fur and meat production, the species got realized and naturalized in the wild in 1999. By 2009, the species spread widely in southern Korea, and Ministry of Environment officially listed it as an invasive species. Its range is currently expanding to the north and causing substantial damages in the natural ecosystem. The primary goal of the study is to provide a genetic basis for targeted management of these notorious invasive species. To achieve the goal, I examined genome-wide genetic diversity and population structure and estimated both census size and effective population size. For sampling between 2021 and 2023, I and my team captured 389 individuals using ripe traps across 17 populations. Muscle tissue samples were collected from across the middle and the lower Nakdong River basin. Here is the process of molecular marker isolation. Given the limited time, I will not talk details. Please just see this flow chart and ask me if you have any question after the talk in person. Briefly, I extracted genomic DNAs and prepared libraries for LASIK, then sequenced the prepared libraries on Illumina HiSeq platform. This slide shows what LASIK is and how I prepared libraries. LASIK is a great advanced molecular method produces thousands of genome-wide markers for a relatively cheap price. But I'll skip it for now. Please feel free to talk to me if you have any question on this molecular method. This explains how I code genome-wide SNP markers with the LASIK data. The genetic diversity measures such as allelic richness and expected and observed heterozygosity were collected. I also estimated inbreeding coefficient and for population divergence as pairwise FST. For clustering pattern of the samples, PCOE and structure will apply. For population size, I use a local census estimated for year tag. Capture marker capture and 3 Poisson equals per box, and using the recurrent Peterson and supplement methods. Then I estimated effective population sizes of regional populations and the Korean populations as a whole using the same growth tool. This is the result of genetic diversity estimation. At the quality filtering, I used over 4,000 SNPs for 239 individuals. Overall, the genetic diversity is rather high, which is surprising for invasive species commonly experiencing genetic diversity. The mean expected heterozygosity was about 0.4, and mean allelic richness is 1.7. Genetic diversity varied across regional populations, as you can see in the table here. Genetic differentiation between populations was generally low, except for a few population pairs associated with GS and NG populations. The average FST across all pairwise comparison was about 0.1. Several population pairs showed FST values across the zero, meaning that these regional populations are not genetically divergent. I plotted the first two major pieces, PC1 and PC2, which explains about 5% and 3% of the total variance. In the plot, you can see two major clusters, Daegu and Busan, separated mostly along the PC1 axis. Based on delta K, the best number of genetic clusters, K was 2. The best case showed that Nigeria population split into two main genetic clusters consistent with the PCOS result. I will explain the detailed clustering pattern in the next slide with a map. Here, each population is shown as a pie chart. The circle size reflects sample size of each population. The pie slices present the proportion of each cluster within that population. 9 population marked with a red box belongs to the Busan cluster, which is dominated by red ancestry. In contrast, cluster 2, the Daegu cluster includes a relatively small number of regional populations. The Daegu cluster is showing green. The effective population size of Korean nuclear population is about 20,000, which is quite large. Between the two genetic clusters, Busan cluster showed much larger effective population size. The Busan and Daegu Gumeo River cluster differ substantially in effective population size, with Busan locally 5 times higher than Daegu. This suggests the Busan populations may represent the key source for regional persistence. The estimated census population size from Chapman method was 170. This value was estimated for a very mixed local region in Busan cluster. If I apply this value to whole distribution area, the census size will be very large. Considering this and the prior information about census size from the documented records, the effective population size I calculated from the genetic data roughly aligns with the census result. However, this result should be interpreted with caution. So, what does this tell us? First, despite potential invasion, Protolac's genetic diversity was well maintained. This mild or lack of diversity reduction can be explained by the large effective population size estimated from this study. The large effective population size indicates that the population maintains a large breeding force even under continuous river pressure. Second, Korea Eritrea has been genetically diverse, forming two genetic clusters, Busan and Daegu. Busan with much larger effective population size likely a key source for regional persistence. Finally, for management, isolated local removal may not be enough if source populations continue to support recolonization. So I suggest treating these two genetic clusters as candidate eradication units. Thank you for your listening.